Hello and welcome everyone. As we have seen, there are different ways of classifying the receptors. The most important is the mechanism by which they act, and that is called as transducer mechanisms. So different mechanisms by which the receptor works. On that basis, the receptors are classified basically into the four types. One, that is G protein coupled receptors. Second, that are ion channel receptors. Third, there are transmembrane receptors, and fourth is intracellular receptors. So let's see about G protein coupled receptors. As the name suggests, these receptors are coupled with G proteins. Now, these receptors are having 7 transmembrane spanning helicals, as it's been described in this figure, and it is coupled with G protein. This is transmembrane receptors, which is situated in between the membranes and associated with G protein. G protein is having 3 different subunits, that is alpha, beta and gamma, and it is associated with GDP. So this is a state of G protein coupled receptors. Now we need to understand about resting phase and activation phase of this G protein, and it is been described in this figure in different steps. So this first step here is the resting state, where there is a G protein, the spanning helicals, a G protein having 3 different subunits, and with alpha subunit that is attached GDP. We are having our agonist molecule here, and it's about to bind to the receptors. So in step 2, our agonist molecule binds to the receptors, and now the process starts. What happens in step 3? Here, because of binding of this agonist to the receptors, now GTP comes into the picture. What happens in step 4? Here, GTP replaces the GDP. So GDP is been thrown out and its place is been taken by GTP. And fifth step is the most important step, and this is a step of activation, where this alpha GTP complex gets dissociated from beta and gamma subunit, and this alpha GTP having its GTPase activity, which produces energy and initiates the response. After the response has been carried out, now the receptor needs to come back to its resting state. So here this GTP is now again replaced with GDP in step number 6, but still it is yet to bind to its original place with beta and gamma subunit. So it comes again into the step 1, and now this GDP again binding with alpha subunit, and alpha subunit binds with beta and gamma and this receptor. So we have already discussed about the activations and resting state of G proteins. Now we are going into bit detail about these G protein coupled receptors. So these G protein coupled receptors acts via 3 basic pathways. Let's say for an example A, B and C. The pathway A is called adenyl cyclase cyclic AMP system, and this we will see. So A we will see, that is adenyl cyclase cyclic AMP pathway. What happens? Our agonist binds with this G protein, and that stimulates our adenyl cyclase. So binding of agonist to the G protein will create either stimulation or inhibition of this adenyl cyclase depending upon the characteristic of receptors. Let's say for an example, in this case we are stimulating this adenyl cyclase enzyme. So what's the role of adenyl cyclase? Its role is to convert the ATP into cyclic AMP. So cyclic AMP has been formed, and it is very important because it is called as secondary messenger. So this cyclic AMP is responsible for the different actions of that receptors. And here in this case, we are taking examples of beta receptors. So this cyclic AMP stimulates the protein kinase A, and it carries out influx of calcium, and because of that the contractility of heart muscle increases. Another function of it is relaxation of smooth muscles. So this is an example of beta receptors, which are G protein coupled receptors which acts through adenyl cyclase cyclic AMP pathway, and especially stimulating this adenyl cyclase. So receptors which stimulates this adenyl cyclase and increases the level of cyclic AMP, these are called as GS type of G protein coupled receptors, and receptors which inhibits this adenyl cyclase and decreases the level of cyclic AMP, these are called as GI type of G protein coupled receptors. Right, so now you know two different types of G protein coupled receptors. These are GS and GI, depending upon increasing and decreasing the level of cyclic AMP, and the response is generated. Let's remember few of the examples of this G protein coupled receptors, especially GS and GI type. So beta receptors, H2 receptors, D1 receptors, all are GS protein coupled receptors where they stimulate the adenyl cyclase, while alpha 2 receptors, M2 receptors, and D2 receptors are example of GI coupled receptors which inhibits this adenyl cyclase. So these are some of the examples you need to remember. So in first pathway, that is adenyl cyclase cyclic AMP pathway, we have seen two receptor, GS and GI. So that is one way of action of this G protein coupled receptor. Let's talk about another way of action of this G protein coupled receptors, and that is phospholipase C IP3-DAG pathway. So in short, second pathway of this G protein coupled receptor is IP3-DAG pathway. Now as the name suggests, phospholipase C IP3-DAG pathway. Here again we'll go ahead with our agonist which binds with G protein, and here only one type of G protein is there, and that is GQ type of receptors. So here initial pathway we had GS and GI, and in this IP3-DAG we have GQ type of receptors. So pathways through which these G protein receptors work, on that basis the name has been given. So here the GQ receptors binds with our agonist and it stimulates the phospholipase C as the name suggests in a pathway, and it stimulates the phosphatidyl inositol diphosphate, and which eventually generates two important secondary mediators or secondary messengers, that are IP3 and DAG. IP3 is called as inositol triphosphate and DAG is called as diacyl glycerol. So these are two secondary messengers in this system. This IP3 is responsible for release of calcium and producing contractions of muscles, and DAG by activation of protein kinase C, phosphorylation process is being carried out and eventually response is generated depending upon the characteristic of receptors. So this is a IP3-DAG pathway, and let's take some of the examples of this receptor. Again, IP3-DAG pathway receptors are called as GQ type of receptors. Alpha 1, H1, M1, M3, all those stimulatory kind of receptors comes under the category of GQ type of G protein coupled receptors. So now we have seen two different pathways, one that is adenyl cyclase cyclic AMP, second pathway that is IP3 and DAG pathway. Let's talk about third pathway, and this third pathway is called channel regulations. There are certain channels which get regulated, which get opened, which get closed depending upon this associated G protein. So this G protein helps in opening and closing these channels and producing a particular response by movement of these ions. And these are some of the examples, like beta receptors helps in opening the calcium channels, and alpha 2 and M2 receptors opens the potassium channels. So by three different ways this G protein coupled receptors produces its action. So now we have completed our first receptor, that is G protein coupled receptor. Hello. So we have already discussed about our first type of receptors, that is G protein coupled receptors. Now we are going into the second type of receptors, and that are called as ion channel receptors. Now, as you know, there are certain ion channels in the body which, depending upon opening or closing, produce its flow of movement of the ion, and due to that produces the response. So how this ion channel works? This is an example of an ion channel which are having different subtypes. For an example, alpha, beta, gamma, delta. So there are different subtypes of or subunits of that ion channels, and in between those subunits there are sites of binding of ligand or the agonist. So these are the sites of binding of the agonist. First, so how the response start is, first there is binding or affinity of the ligand or the agonist to the receptor site, and on binding this ion channels gets activated. So let's say, for an example, these are of flower shape and they gets activated. Upon activation, these channels open up. For an example, this is resting state or inactivating state, and this is activation state. So on binding of ligand, channel becomes open, and now there is movement of ion in and out of those channels. Depending upon the site and the characteristic of the channels and the affinity to the particular ions, there are movement of those ions in and out of the cell, and eventually that produces the response. So these are ion channel receptors, quite easy to understand. Important thing is these are the fastest acting receptors. This receptor acts the fastest. These are multiple time asked MCQ. Which is the fastest type of receptors? So this is the fastest acting receptors. Which are example of these ion channel receptors? Nicotinic receptors and GABA A receptors that you need to remember. So this was simple second type of receptors, ion channel receptors. Let's go to third type of receptors. These are transmembrane enzyme linked receptors. So as the name suggests, these are transmembrane receptors which are having outside portion which is agonist binding portion where the agonist usually binds, and inside the cell there is a portion where tyrosine residue is being attached, and this is a site of enzyme, because it's been said it's enzyme linked receptors. So this is a site where enzyme works. Now in this case, which enzyme works? In this case, tyrosine kinase enzyme works. So upon activation of this receptor by the agonist molecule, now there is a process called as dimerizations, where the two identical receptors comes closer to each other, and there is a process of phosphorylations by the enzyme tyrosine kinase of this tyrosine residue, and due to that phosphorylations the downstream signaling process goes on and eventually response is generated. So which are the molecules that acts through this transmembrane enzyme link receptor or tyrosine kinase receptor? These are insulin, very important, growth factors, and atrial natriuretic peptide. So these are some of the examples of the endogenous substances which acts or exogenous substances which acts through this transmembrane enzyme linked receptor where the enzyme is tyrosine kinase. Another enzyme associated with this kind of transmembrane receptor is Janus kinase. So these are again the same kind of receptor where there is outside the cell there is agonist binding molecule, but inside the cell in place of tyrosine there is activity of Janus kinase enzyme, and on activation with agonist after the dimerization status there is Janus kinase activation is being carried out. But here one more step comes into the picture, that is role of STAT. Now what is STAT? STAT is basically a signal transducer and activator of transcription. The STAT is inside the cytoplasm. By activation of Janus kinase, this STAT comes in contact with Janus kinase and eventually the process of downstream signaling is being carried out. So earlier in the case of tyrosine kinase, there was only one enzyme that was involved, tyrosine kinase. Here Janus kinase is involved, but Janus kinase activates this STAT and produces its response. So there is an important mediator, that is signal transducer and activator of transcription, and eventually by transcription it produces its response. So substances which works through these mechanisms are hormones like growth hormone, prolactin, and some interferons we'll see in antivirals works with this Janus kinase STAT system. So this is also a kind of same transmembrane enzyme link receptor. This was also the same, but here the enzyme was tyrosine kinase and here the enzyme is Janus kinase. So these are third type of receptors. These are transmembrane enzyme linked receptors. And now we are heading into the fourth type of receptors, and these are also called as intracellular receptors because these receptors are present inside the cell. Now where inside the cell? This receptor may be present into the cytoplasm of the cell or they might be present inside the nucleus of the cell. So depending upon their location, they've been divided into intracytoplasmic receptors or nuclear receptors. These receptors are slowest acting receptors. We have already discussed which are the fastest acting. Yes, ion channel receptors are fastest acting, and these intracellular signaling receptors are slowest acting receptors because there are multiple steps involved logically and it takes time to produce the action. These are also called as receptors regulating gene expressions because they will be acting on transcription and eventually they will act by forming the specific signaling mRNA. So best example is glucocorticoid receptors. Glucocorticoid is a hormones and its receptors are basically these intracellular kind of receptors. So let's take an example of these glucocorticoid hormones which act as an agonist. But here the receptor is not on the cell membrane, receptor is inside. So these glucocorticoid receptors are present inside the cell and they are usually attached with some of the proteins. Here it is called as heat shock protein. So there are multiple proteins with which these glucocorticoid receptors are attached. So this agonist goes inside the cell and gets attached to a particular site on the receptors. Now this receptor goes inside the nucleus and produces dimerizations, and depending upon the activators or repressors associated with this protein or receptors, the process goes on. So if co-activators get activated, the transcription process gets activated, mRNA is being formed, and the particular messenger RNA which is being formed, which helps in protein synthesis, and eventually there is a response which is modification of cellular functions. So this is how this intracellular signaling receptor works. Because there are multiple steps involved, these are slowest acting receptors, and we have already divided these receptors into two parts depending upon their location of receptors. So if their receptors are in cytoplasm, these are called as cytoplasmic receptors. So substances like progesterone, androgen, glucocorticoid, and mineralocorticoid, they work through these cytoplasmic receptors, while some of the receptors are present inside the nucleus, and these are called as nuclear receptors. And hormone like estrogens, some vitamins like vitamin A and D, and hormone like thyroid works through these nuclear receptors. So now we have seen all these four types of receptors. These are one that was G protein coupled receptors, it having three different pathways: adenyl cyclase cyclic AMP system, second IP3DAG system, and third ion channel regulation. Then we saw the second type of receptors, that are ion channel regulated receptors. Then we saw third kind of receptors, these are transmembrane enzyme link receptors. And fourth was intracellular receptor. So we have completed different type of receptors now.